know, not all the eukaryotic genes transcribed will be translated into proteins. This is due to a process called RNA splicing. RNA splicing is a process that removes the intervening non-coding sequence of genes or introns from pre-mRNA and joins the protein coding sequence or exons together in order to enable translation of mRNA into protein.
Borders between introns and exons are marked by specific nucleotide sequences within the pre-mRNAs. These sequences describe where the splicing will occur. 5' splice site of pre-mRNA includes a GU dinucleotide at the intron end. 3' splice site region has 3 conserved sequence elements, which are adenosine at branch site, polypyrimidine tract, and AUG terminal. Spliceosome assembles on these sequences and catalyzes the splicing reaction.
Splicing is catalyzed by spliceosomes. Spliceosomes are composed of small nuclear ribonucleoprotein sRNP, which are U1, U2, U4, U5 and U6. Some non-sRNPs also involved in splicing. For example, U2 auxiliary factor U2AF recognizes the polypyrimidine tract splice site, and in the initial step of the splicing reaction helps another protein, branch point binding protein BBP, bind to the branch site adenosine A.
Initially, the 5' splice site is recognized by the U1 snRNP. The 65 kDa subunit of the U2AF binds to the polypyrimidine tract, and the 35 kDa subunit of U2AF binds to AG at the 3' splice site. The former subunit interacts with BBP and helps protein bind to the branch site. This arrangement of proteins and RNA is called the early E complex. U2 snRNP then binds to the branch site adenosine added by U2AF and displacing BBP. U2AF then leaves the complex. This arrangement is called the A complex. U4 and U6 snRNPs along with the UP snRNP join the A complex. With the entry of the 3 snRNP, the A complex is converted into the B complex.
Then U1 snRNP interaction at the 5' splice site is replaced with U6 snRNP, and the U1 and U4 sRNPs are released from the complex. This arrangement called the C complex that produces active site which catalyze the 2 esterification step of the splicing. In the first trans-esterification reaction, the 2' OH of the bulk A residue at the branch point attach the phosphate at the 5' splice site. This leads to cleavage of the 5' exon from the intron and the ligation of the 5' end of the intron to the branch point to 5' forming a lariat. In the second trans-esterification reaction, the 5' OH of the detached exon attach the phosphate at the 3' end of the intron. This leads to ligation of 2 exons and release of the intron still in the form of a lariat. The spliceosome complex is subsequently released from the lariat intron and disassembled. The lariat is debranched and degraded.